Okay, so just very briefly, it hardly needs to convince you guys that this is an interesting question, but just to remind ourselves at the top level, it's just plain interesting. I mean, who hasn't spent a lot of time looking at faces and just marveled at the richness and heterogeneity and evocativeness of faces, right? I mean, Isabel mentioned that people used to study faces as a window into the personality, but I think that's part of why they're so fun to look at, is we feel like we can see all that stuff in people, as some of the recent literature is now testing. And I think also when we catch ourselves making mistakes in face recognition and failing, it's also just somehow inherently captivating and interesting. And the difficulty of describing faces also makes them very interesting. And finally, the individual differences. People really differ greatly. As recent work from Ken's lab and mine has shown, those individual differences are large and stable. They're heritable. And they are not correlated with IQ. I'm happy to report, because I'm particularly bad. Another reason to study face perception is as a potentially more tractable case of object recognition, right? If you're trying to, say, characterize IT cells and how are they tuned for shape, it's just hard to even figure out how to get started. There's so many different kinds of shapes. But at least with faces, you can imagine a finite list of features that might matter. And so many people have studied faces as a sort of test case for object recognition. Almost the opposite argument is to look at faces as a special case. And so there's this long tradition in especially cognitive psychology to consider the idea that much of cognition is conducted by these domain-specific mechanisms that do very, very specific things, from Chomsky's work on language to more discussion of this topic by Fodor and many others. So these last two notions are really kind of at odds, right? To the extent that face recognition is a domain-specific special case, then it doesn't inform us about the general case. So I think they're both interesting, but they kind of can't quite both be true. So there's some tension there. So on this notion of specificity, Isabel's already mentioned there's evidence going way back from prosopagnosia. Actually, on some reports, the papers go back to the 1800s, but this is the first one that was really widely cited, Bodimer in 1947. I think even more interesting is the opposite case. There are a couple reports. The best one is by Moscovich et al. In 1997, a patient with severe agnosia who was just extremely bad at recognition of objects and words and scenes and pretty much everything, but 100% normal at face recognition. And that, if you think about it, is even more striking evidence that face perception engages different machinery from object recognition. The fact that you can blitz pretty much the whole system except for face recognition. Josh has a question. Everything. Yeah, he did all kinds of different stuff, including familiar and unfamiliar. Isabel also mentioned the behavioral evidence that face recognition is special. My favorite is the study from Yin on the inversion effect. Here's Yin's data here from 1969. This was his PhD thesis in this department. And what I love about it is that it's the lowest tech thing you can imagine, right? He just gave people faces upright for study and tested them upright, or they studied them inverted and tested them inverted. And he found that when you did this with houses and stick figures and other things, people did worse when they studied and tested stimuli inverted for all classes of stimuli, but that inversion cost was much greater for faces than for anything else. And he actually speculated, based on that humble behavioral data, that there might be special brain mechanisms for face recognition. I think that's pretty cool. And Bob's data, he's already described here. But so there was heaps of prior reason to think that face recognition was interesting and maybe special from the early work. But what I think those earlier lines of work really left unanswered were a whole bunch of questions, from the basic functional neuroanatomy in normal subjects, right? So all of this stuff was either done in monkeys or done in people with brain damage. And so it was really hard to, lesions are large. It's really hard to tell what the configuration was in the brain in normal subjects. And this question of specificity, as Bob said, there were problems going back and finding that same cell to do repeated testing, and there was much left unanswered about precisely how specific those mechanisms were. It wasn't known whether there were one or a whole set of different brain regions for face recognition. And most interestingly, the the representations of faces and the computations that go on in face recognition were really pretty much unknown. So brain imaging offered the first ability to potentially get a direct measure, a direct readout of the neural signals involved in the face system in typical human subjects. So the first brain imaging study, I think, that looked at faces is this paper from Jim Haxby and Leslie Ungerleider and others in 1991. They scanned people with PET while they did a matching task. They had to say which of those two faces on the bottom matched the one on the top. Or they had to do a location matching task. They had to say which of these two dots was in the same place as the one in the upper display relative to the double bar. And if you look at it for a minute, it's actually kind of hard. I mean, you can do it. It just takes a moment. And each of those was compared to a sensory motor control where subjects saw empty boxes and just alternated their finger presses left, right. And here is what imaging data looked like circa 1991. So the finding is that up here, oh boy, it's worse with this projector. There's a few kind of light boxes up there, which were the activations for the dot matching compared to the sensory motor control. And down here, the darker ones this is a face matching compared to sensory motor control. And the basic point of this paper was that you got a difference in location tasks and face tasks, which are supposed to echo the what-where distinction found previously in monkeys.
Was this the whole brain color?
I think it was a whole brain. You can't see it, but these things go way up to here, either the back half of the brain or the whole thing. Yeah. Okay, so what's nice about that is PET offers this absolute measure instead of these numbers that are sort of meaningless that you get with functional MRI, like, signal intensity of 623, which means exactly nothing, right, except in comparison to something. With PET, you actually got a measure of what percent did the blood flow change between conditions, and it was large, 30% differences shown here in blood flow in these conditions. So that's cool. But in other ways, there are obvious limitations here. The resolution is really low. And importantly, you couldn't rescan subjects because the radiation dose of participating in one of these studies, that was it for the year. So you couldn't put the same subject back in. And so it's very interesting what Bob was saying about the fact that you couldn't find that same cell. I think in the early days of functional imaging, the very same problem was really at the root of inability to drill down and understand functional activations with PET. You couldn't go back and find that same stuff, right? Anyway, this study didn't test face specificity by design. It wasn't intended to. It was just the first paper I found that had faces as stimuli. First one that really tried to look at face specificity was this paper by the brilliant Justine Herschel. I mean, I don't love this paper, but I think she was brilliant in all kinds of ways. I mean, the paper was heroic for what it did way back in 1982. They had only 7 subjects, and they had what I think was the right idea, scanning subjects, looking at faces and looking at objects and doing a bunch of different tasks. But this paper, even though it's often cited as finding evidence for face specificity, did not find any brain regions that responded significantly more strongly when viewing faces passively compared to passive viewing of gratings, or when doing recognition tasks on faces versus objects. They got nothing. Actually, I think that's not so surprising for a bunch of reasons. One is it was extremely underpowered with only 7 subjects. And the other is, as I'll mention in a moment, this is a group analysis. And even with functional MRI, with the best of circumstances, you usually do not see face-specific regions with group analyses. And I'll discuss why in a moment. Okay, so after this, there are a few more PET studies, but the important event was when functional MRI came on the scene in the early 90s, and this is one of the first, possibly the first, functional MRI study looking at face processing. This is by Ina Puce and Greg McCarthy and Trude Allison, and they scanned people with a nice block design while they were looking at faces or letter strings or textures, and they found stuff like this. So here's a coronal slice with 2 blobs in the right hemisphere that responded more when subjects were viewing faces like these or letter strings like those. And in the left hemisphere, they found a little blob that responded more to letter strings than faces. So now we're starting to get somewhere. That's some evidence for specificity, but if you look at these stimuli, you can see all kinds of ways they differ. So it's not very strong evidence for specificity. It could be any number of things from just retinotopic extent to the processing of generic shape or any number of other things. So it's around here that I decided to jump into the fray. I meaning me and Josh McDermott and Marvin Chun. Here we are, circa 1996. It really was a pretty wild time then. I had been actually trying to claw my way onto an imaging machine for literally 10 years before I finally got to scan. I got an email from Bruce Rosen one morning, and he said, okay, Saturday morning, 6 to 9, the scanner's yours. And I was at once thrilled and a little freaked out. I had a long-distance relationship at the time. My partner, John, over here, lived in D.C. We saw each other only on weekends. And John said, okay, you have to choose. It's me or the scanner. So I made a gamble, and I said, okay, it's the scanner. And he buckled. I knew he would. Anyway, it was not the most convenient time, but what can you do? So Josh and Marvin and I would get up at 5 in the morning and drive over to the scanner, and it really was very wild. I remember actually a lot of hilarious things about it. I remember Marvin was just not very good at getting up early. He lived in an apartment in Harvard Square, and I would park right next to Out of Town News, right in the middle of Harvard Square, and get out of the car and scream at the top of my lungs, Marvin, wake up! And eventually he would wake up and we'd drive over to MGH, where the way you got into the scanner room was nobody had a key. It was just not organized. There wasn't a system. There was no kind of safety controls, no nothing. Everyone who scanned there took their ID card and just kind of wiggled the lock to get in the door. That's how we got in. And then when we got in, we had no idea what we were doing. Absolutely no idea. There was no one to show us. And so we'd just go over there and we'd wing it. And you had to shim the scanner for over an hour. It's all automated now, but at the time, you just fiddle with all these things. It was a total black art. We had no idea what we were doing. There was just this big peak, and you needed to get it narrow. We didn't even know it was a spectrogram. But anyway, very often what would happen is after we finally shimmed the scanner, a message would come on the screen, and it would say, "Arcing may occur." And the first time it happened, we were really frightened. But after a while, we knew that when the arcing may occur message came on, we were golden. We were good to scan. Anyway, so, another thing that happened is the scanner used to break down all the time. And we'd be so scared because it's like this $3 million thing, and we're total schmoes. We have no idea what we're doing. It would break down, and Josh McDermott, who I think was 19 at the time, would very calmly stroll over to this bookshelf that had about 20 volumes of instruction manuals on how to operate the scanner. And he would just calmly start pulling down volumes. And I'm just like, oh no. My strategy was to run into the next room and find Ken Kwong, who pretty much invented functional MRI. And Ken was always at the scanner, and he would come in and he would say, oh my, you broke the scanner. Let me see if I can fix it. And he would, always. He was great. Okay, more seriously, what do we do? I was actually more interested back then in object shape. As I said, I think that's the general case of visual recognition. Why would you study the weird exception case if you could study the general case? So I was looking at the processing of object shape, and we were kind of sort of getting some stuff. We were contrasting what happened when people looked at familiar objects versus line drawings of possible but unfamiliar 3D shapes versus scrambled stimuli. And we were finding various bits of brain that responded a little bit more in some subjects when you looked at shapes versus scrambled junk. And that seemed sort of promising, but it just wasn't very reliable. It was pretty weak, and it kept going away. And after a few months, I had no money and no support for the scanning, and I knew if I didn't get a result fast, I'd get kicked off the scanner. So at a certain point, I said, okay, I don't know what's happening with this. We're going to pull the plug on this, and we're going to study faces because all of the prior literature says there must be a honking big signal in there, and we need a honking big result, and so we'll do faces. So we did the screamingly obvious thing of scanning subjects while they looked at faces versus objects like this. Nice simple block design. And we scanned subjects doing this simple paradigm. And here's one of the first scans on me doing that. And what you see looking at this is a bunch of horizontal slices through my brain. And that little region right there is the bit that responded more when I was looking at faces than objects. And if you dumped out the time course from that region, you saw stuff like this. A nice, big, clean, higher response to faces than objects. So that seemed pretty good. And we saw this in most of the subjects we scanned, so we felt reasonably good about it. So I presented this at 2 different conferences in 1996, the Human Brain Mapping meeting. It was its second meeting in Boston. And when I presented it there, I put up this slide, and Leslie Ungerleider said to me, "How can you really have only that one spot? You must have thresholded it out all your other activations or masked them out." And I said, "No, look at my significance bar. It goes all the way down to p less than 0.01. And she was pretty surprised. No one believed that you could get anything this spatially precise. When I presented this same slide at the Society for Neuroscience later that year, Ed Rolls came up to the microphone, as he is wont to do, and he said, "This can't be true, because when we record from the temporal lobe in monkeys, we never find any patch that has higher than 20% face-selective cells." And I said some slightly wimpier version of, "Well, you must be recording in the wrong place, Ed." I didn't quite say that, but almost. In any case, it was, people did not want to believe this. That was considered pretty wacky. Okay, there are a couple of issues here. One is the statistics were a real problem. So this significance bar is not corrected for multiple comparisons. There are tens of thousands of voxels in this study, and so you can't take the p-levels seriously, and that posed a problem. Bonferroni corrections would take you down to about 10 to the minus 4. This little blob would still survive, but you don't want to have to always find things at 10 to the minus 8 significance to be able to believe anything. This guy, Carl Friston, at the time was starting to publish things about what was the right, less conservative less restrictive kind of correction, but it was hidden in a lot of esoteric math, as Carl Friston likes to do. And I didn't understand his math, and I didn't want to trust his math, so I was in a pickle. And I just said, okay, the first thing that comes to mind is I'll just do this. Here's what I did. I scanned myself again and again and again and again. So here's a paper that I published in 1996. This was a conference proceedings, and so there weren't any reviewers, and so we just kind of basically published our pretty raw data. And I said, look, here's subject one, that would be me, on 6 different scans, and there's a really similar blob in all of them. So I'm not really sure what to do with that p-level issue, but whatever it is, it's real. I think Ken advised me to do this too. He was very helpful back then. So that convinced us that some of this stuff was real. But that's not a great solution. What are you going to do with other subjects? You can't have a whole field based on one subject. That's kind of dysfunctional. And the problem here is that there's anatomical variability. So here are 12 of our first subjects. And you can see, here's me with my fusiform face area, and here's another one, and here's another one. And there's a family resemblance. They're similar, but they're not in the same place. And that's why if you do a group analysis, you don't usually see much. And it's a problem. So what are you going to do? So we sort of thought about it and decided that the thing to do is find that region in each subject individually with a localizer scan and then study it. So we'd scan someone on this basic thing I just described, faces versus objects. We'd find a blob. I believe this is Randy Buckner, circa 1996. And we'd circle those little face-selective voxels. We'd look at their nice time course. And then having found it, we would ask a new question and say, okay, are you really a face area? Or do you respond to any human body part? Do you just respond to interesting things that you're paying attention to? Do you respond to only front views of faces, et cetera? You could test all those kinds of things by just running some new conditions. So here are some of Ken's stimuli and some of his students. And we scan people doing a comparison, a matching task on faces versus hands. And the idea was that hand matching is actually very difficult, harder than face matching, and so we were forcing subjects to attend more to the hands than the faces. And when we got out pretty much the same face-selective blobs and higher responses to faces than hands, we could rule out all 3 of those alternative accounts. It wasn't just any body part, it's not just attention, it's not just front views of faces. You can also see we didn't even know about drift correction back then. We just kind of took the raw data and dealt with it. Okay, so that method solves a whole bunch of problems. It kind of prevents you from doing this sort of dysfunctional thing of kind of sniffing around in your data for some activations that look good and then publishing them. That's dangerous. There's too many degrees of freedom. It locks you down by saying, okay, you got to find the region you care about and the criterion you're looking for in advance, lock everything down, and then test that region on new conditions. So you have built-in replications and cross-validations, and in this era when everybody's worrying about the replicability of science, it's nice to have a method that has a built-in replication. And because you're not doing multiple comparisons when you find one region of interest, you don't need to correct for multiple spatial comparisons. Instead of testing 50,000 voxels, you're testing one ROI. So it gives you a huge boost in statistical power, and it handles that variability in the anatomy across subjects. Plus, importantly, it gives you a nice unbiased measure of effect size, right? So you have left out data and you just measure the magnitude of response in different conditions. You can take those data as a really nice measure of the magnitude of that region. Okay, so here's our first paper using this method, our first real paper, where we used this method on a bunch of different experiments and argued that that region seemed to be pretty selective for faces. As a sidebar, this entire paper was analyzed in Excel. I'm not kidding. I guess SPM existed, but I didn't know about it. I didn't have anybody to tell me about it, and I didn't know what I was doing. So I just pulled out those raw time courses and I pulled them up in Excel and I averaged the time bins myself by hand and wrote them down on a piece of paper and averaged them in a calculator. Proceeding in this fashion over the first few studies, some of which Ken was involved in, we found basically if you looked at the response of that region to lots of different conditions, it responded to a wide array of different kinds of stimuli, all of which you could call faces. So as Bob mentioned, for the case of face cells, really seems like, if the stimulus looks like a face to us, that thing's going to respond. The same thing was true of the fusiform face area. And in contrast, lots of different kinds of control stimuli produced a much lower response. And my favorite contrast here is the Mooney faces when inverted. Almost nobody sees a face there. You take the same stimulus and you present it upright, and people almost always see the face, and we got a much higher response in the upright than the inverted case. And that reassured us that it wasn't some low-level property of the stimulus. Oops, wrong direction. Okay, so this thing is present in pretty much every normal subject. It's pretty selective, but that's just the barest beginning. At this point, all you can do is say, okay, there's a bit in there, it responds selectively to faces. From there on out, there's a huge number of questions, and I think the progress with neuroimaging is, real. There's some, but it's measured, and oops, what? I think it just crashed is what happened. Right, hang on. Yeah, I should use keto. All right, oh no, I don't want to do this because now I can't skip ahead. There. Okay, so one wants to know about the broader architecture. I've only been talking about this one region so far. So other things that have been found, so there's both this fusiform face area, but there's another region, sometimes called the occipital face area. And so, you can see both of those in most subjects. Whether those are really different beasts, people tend to assume they are. I'm not so sure. Really, to know, we'd have to be much more careful to look at the region in between, find out whether the very subtle functional differences between those regions are greater than the gradations you see within. None of that's really been done very carefully. There are reports of an anterior face region up here. Many people see them. I don't see them regularly enough that I know how to study them, which is too bad because I think that would be a promising place to look. Another region that people have, dozens of papers have talked about the specificity of this, the face specificity of this region in the SDS, but Bendine has recently shown that it's not a face area at all. It responds at least as strongly when you listen to voices. So we don't know what it's doing. Ben's done some very interesting studies of that region, but it's not a face area. Another region that responds quite strongly to faces is LO, lateral occipital complex. It responds more to intact than scrambled stimuli, but it also responds a lot to faces. So it's a potential other actor in this system here. So we sort of have a sense of who the actors are, but how does each of these regions contribute? There's some data, but I would say less. So really what you need is causal tests. And these are hard in humans. We basically have two methods, both of them sparse. You can use TMS, and TMS has shown that if you zap that occipital face area right there, you interfere with face discrimination. You do not affect discrimination of objects or bodies or other stimulus types. So that's nice evidence that that region is not only causally involved in face perception, but selectively causally involved. That's nice. Unfortunately, you can't zap all the other regions. They're too medial. So you zap the ones you can. LO, if you zap LO, what you find is you disrupt perception of inverted faces, not upright faces. And that's consistent, yeah. Yeah, I published it. Okay. Me and David Pitcher did that. Yeah, so if you zap the occipital face area, you disrupt perception of upright faces only, not inverted faces. And if you zap, actually both. But if you zap LO, you only disrupt inverted faces. And that's consistent with the idea that an inverted face is processed like an object, right? Similarly, that patient CK I mentioned, who's severely agnosic but normal at face recognition, his behavioral face inversion effect is 5 times greater than normal, presumably because he's only operating on that system, on the face system, without LO to contribute. The other method is electrical stimulation of these regions. This is very rare. The data are precious. If you haven't seen the Parvizi and Grill-Spector video of the patient being zapped in their fusiform face area, ask me later, I'll show it to you. It's quite remarkable. And it's 2 minutes of data, but what it shows is that zapping that region distorts the percept of a face quite radically. And with the small amount of data they have, it doesn't seem to have much effect on perception of other stimuli. So these causal data are precious, but they're not enough to go on. We really need more systematic causal data, and they're really hard to get in humans. Obviously, we also want to know what the representations are in there, and what computations are conducted in each of these regions. And there, I'd say we've made some progress, but it's not very satisfying. So we have basically 2 methods in humans with functional MRI. Functional MRI adaptation, which has told us some stuff about representations in the fusiform face area, and the other face regions. So, for example, we know that there's representation of individual identity because you get identity-specific adaptation for faces in the FFA. You only get it when the faces are upright, not when they're inverted. So that's nice and checks with the behavior. That's nice, but it's kind of step 1. And from there on out, there's a few other findings, but they're hard. The effect sizes are small. It's a fiddly business. The other method that's sometimes useful in functional MRI in characterizing representations is multiple voxel pattern analysis, where you look at the profile of response over all the voxels in a region. And that, I was very excited when it first came on the scene, but one of the first things I did was to scan a bunch of subjects with the highest resolution we could get, 1 millimeter voxels. We scanned them from several subjects for 2 hours, looking just at pictures of Harrison Ford or Julia Roberts. And we could not decode from the fusiform face area which face they were looking at. And that was just so disappointing, because you'd think on any story, if there was any useful face information, you should be able to decode it. So since then, a few people have reported very weak ability to decode some kinds of face information from the fusiform face area. It's always just barely above chance. Many people have failed to see it altogether. It's just such a tiny effect that it's really hard to build a field on. So that's pretty unsatisfying, and in fact, it's not surprising, right? Because this pattern analysis method works when cells with similar selectivity are spatially clustered at approximately the grain of voxels or close to the grain of voxels. And for lots of things like that's just not true, right? And so Doris Tsao published, yeah, or a few hundred, yeah. So a voxel's big, right? So lots of things are not clustered like that. Yep. So there's a recent paper from Doris Tsao's lab where they show this very directly in monkeys, where they did functional MRI in one of the face patches in monkeys, and they also did they used decoding to try to decode face identity, and they totally failed from this face patch in monkeys using functional MRI. But in the same face patch in the same monkeys, if you recorded from a bunch of neurons in there, you could decode just fine. And that just shows the information is in there, you just can't see it with functional MRI, which is very frustrating. Of course, we also want to know what circuits are involved in computing these representations. We want to know what these different brain regions are connected to, how they interact with each other. We want to know when and how they develop. That one's really important and really difficult. I've spent the last 5 years trying to answer that. I'm getting almost nowhere. My real view is that all of these questions are really better answered in monkeys. I think in monkeys you can do better causal tests. You can use muscimol, you can use electrical stimulation targeted to specific patches in trained monkeys to really ask what tasks are affected by each of these things, what are they necessary for. You can actually see the neural population code by recording from actual neurons. You can do Vinrick's beautiful study with Sebastian Moeller and Doris Tsao, where they electrically stimulated one of the face patches while scanning with functional MRI in order to see the connectivity of that region, showing that the different face patches are strongly connected to each other and not to the cortical regions in between. Love that study. And to study development, you can in principle do controlled rearing. So I think these are the methods that are really going to be necessary to get at these questions. And sadly, most of them don't work in humans. They only work in monkeys. But that raises my last question, I'm almost done, is how homologous are these regions between humans and macaques? And the good news is that the behavioral data suggest lots of homologies between face perception in humans and monkeys. And in some recent work with Rosa Leifer-Souza and Bevel Conway, we've been looking at the kind of spatial configuration of the face system in macaques and humans. So Rosa and Bevel published this paper a couple years ago where they showed that in the monkey cortex, going down the temporal lobe, you have a sandwiching of face patches, outlined in black here, patches that are somewhat biased to respond more to color, outlined in white, and patches that are responsive selectively to scenes in green here. So it goes faces, color, scenes. In a paper we just submitted, we find the same things in humans. On the ventral surface, we find a systematic adjacency of faces, color, and scenes. It's in a geographically very different location. In humans, it's down on the bottom of the temporal lobe, whereas in monkeys, it's out on the side. But we're making the argument that this whole tripartite system basically got pushed over by language or other functions in there in the temporal lobe and moved down on the ventral surface of the brain, and those regions are homologous. So that's good news. It means that results from using these much stronger methods probably apply to humans. I think there are limits to the grain of this homology. It would be lovely if we could say this face patch in humans corresponds to that face patch in monkeys. I think that's going to be a tall order. We need more than functional similarities. We probably need connectivity and gene expression and God knows what else. But even short of that, I think the idea that these broader systems are roughly homologous means that more progress will come from the monkey research. So thank you.